Hello friends, today I am going to explain about a UV visible spectrophotometer. UV visible spectrophotometer is an instrument, analytical instrument. It is commonly used to measure the amount of ultraviolet and visible light that is absorbed by the sample. It is a widely used technique in biochemical laboratory and it is used for both qualitative and quantitative analysis. Now we will see the different components of the instrument. There are 5 different components. First one is a source of electromagnetic radiation. Then there is a monochromator for selecting a narrow band of wavelength. Third one is a sample cell or a cuvette. Then there is a detector or transducer for converting radiant energy into electrical energy. And finally, there is a device to read out the response of the detector, probably a computer. Now we can see the source of continuous radiation. For visible region, commonly a tungsten filament incandescent lamp is used, and for ultraviolet region, a low pressure hydrogen or deuterium discharge tube is generally used. And for ultraviolet source, there must be a quartz window because quartz is transparent to UV, but at the same time glass will absorb UV region. Second component is a monochromator. Monochromator consists mainly lenses and mirrors to focus the radiation. In addition to that, it has an entrance slit to restrict unwanted radiation and help to control the spectral purity of radiation emitted from the monochromator. And there is a dispersing medium to separate the wavelength. So it will separate into different wavelengths. There are two types of basic dispersing elements we can see in a UV visible spectrophotometer. One is a prism and then second one is a diffraction grating. In addition to that, various types of optical filters are also used in different spectrophotometers. So first we will see prism. When an electromagnetic radiation passes through a prism, it is refracted because of the index of refraction of the prism material is different from that of air. Shorter wavelengths are refracted more than the longer wavelength. So the effect of refraction is to spread the wavelength apart into different wavelengths. Then in the visible spectroscope inside the monochromator, this prism will be in a rotating mode. So different wavelength of spectrum can be made to pass through an exit slit and through the sample. Since it is rotating at one time, particular wavelength can be coming out through the exit slit and it will be passing through the sample cell. Next is diffraction grating. Diffraction grating consists of large number of parallel lines or grooves ruled on highly polished surface such as aluminum. There are about 15,000 to 30,000 such grooves per inch for diffraction of ultraviolet and visible light. These grooves act like scattering center for rays falling on the grating. So we will get a linear dispersion. The figures shows the dispersion of polychromatic light by both prism and diffraction grating. Various types of optical filters can be also used to isolate particular wavelength. There are narrowband pass filters, sharp cut filters and indifferent filters. The first two are usually made up of glass and contains chemicals, especially dyes that absorb all radiation except that desired to be passed. The sharp cut filters absorb all radiation up to specific wavelength and pass radiations at longer wavelength. Next component of the instrument is sample cell. The cell holding the sample must of course be transparent in the wavelength region being measured. So the cell used usually will have 1 cm thick, and for UV region usually a quartz cuvette has to be used because quartz cuvette won't absorb UV radiation. For visible region we can use glass or quartz. Both UV and visible light we can use same kind of detector. So there are different types of detector, for example phototube, photomultiplier tube, diode array, etc. First one is phototube. Phototube is also known as photocell. It is commonly used in ultraviolet and visible region. This phototube consists of a photoemissive cathode and an anode. Then a high voltage is impressed between anode and cathode, usually 50 to 90 volt. When a photon enters the window of the tube and strike the cathode, an electron will be emitted. This electron will be attracted to anode, causing a current to flow that can be amplified and measured. The response of the photoemissive material is wavelength dependent. Hence, different phototubes are used for different regions of the spectrum. For example, one may be used for blue and ultraviolet portion and the second one for the red portion of the visible spectrum. Second one is a photomultiplier tube. Photomultiplier tube is more sensitive than a phototube. It can be used for both visible and ultraviolet region. Photomultiplier tube also consists of a photoemissive cathode which the photon strikes, and there will be a series of electrodes or diodes each at more positive potential 50 to 90 volt than the previous one. When an electron strikes the photoemissive cathode, a primary electron is emitted. The primary electron released from photoemissive surfaces accelerated towards first dynode. The impact of electron on the dynode surface causes the release of many secondary electrons, which in turn are accelerated to next electrode, where each secondary electron releases more electron. This process will continue up to about 10 stages. So, from a single photon, about 10 raised to 6 to 10 raised to 9 electrons will be generated. And these electrons are finally collected by the anode and which is electronically amplified. Third one is diode array detectors. They are mainly used in spectrophotometer that record the entire spectrum simultaneously. Diode array consists of series of hundreds of silicon photodiodes positioned side by side on a single silicon crystal or chip and has an associated storage capacitor that collect and integrate the photocurrent generated when photons strike the photodiode. So, here the entire spectrum can be recorded simultaneously. How a UV visible spectrophotometer works? The light from the source enter the instrument through an entrance slit which is collimated and focused onto dispersing element, usually a diffraction grating. So, the wavelength is selected by simply rotating the monochromator and desired wavelength will be impinged on the sample. The intensity of radiation transmitted to the sample is measured and recorded as absorbance or transmittance. So, see you soon with another video. Thank you.